Okay, thanks Ian for the invitation. So I'll tell you today about our work on neural codes for natural behaviors in flying bats. So I'm interested broadly in the neural base of behavior, but you can't study all of behavior, you have to focus on something. So we focus mostly on two topics, on spatial behaviors, so navigation, spatial cognition, spatial memory, things around space, and also more recently social behaviors, and I'll talk about both of those. And the brain areas that I'll be talking about, which are sort of the stars of this talk, are the hippocampus and the entorhinal cortex here in the brain of a rat. This purple sausage is the hippocampus, and this strip at the back of the brain is the entorhinal cortex. And we know for many years that these two brain areas are important for memory in general and spatial memory and navigation in particular. And when you go into these brain areas, people have found various neurons that seem to represent space. So what do I mean by that? John O'Keefe in the 1970s found in the hippocampus place cells. So what is a place cell? If you let a rat, in this case, run in a square box, about 1 by 1 meter in size. So here you see this square box. There's a top view. The black line is the trajectory of the rat over 20 minutes or so. And you record from one neuron in the hippocampus of the rat. And the spikes, the action potentials of this one neuron are depicted in red here, the red dot. So you see this neuron likes to fire primarily when the rat is in the northeast corner. In this case, you can turn it into a heat map. So whenever this neuron fires, it sort of says the rat is in the northeast corner. And another neuron might fire when the rat is in the northwest corner or in the south, or etc. So together you look at an ensemble of such neurons, and this encodes a map of self-position, a map of where am I in space. So we have neural maps in the brain, and then going to the medial entorhinal cortex, the Mosers in Norway have found grid cells. So these are neurons that are measured in experiments that are virtually the same. Again, a rat running in a square 1 by 1 meter box. There's no real navigational task here. Food is sprinkled randomly, and the rat is just foraging randomly around. But despite this random walk in black here, this neuron, one neuron fires in a set of positions. So whenever the rat is located here, or here, or here, or here, or here, the neuron fires. So this forms a sort of a hexagonal lattice. So this looks almost like a millimeter paper over space, but instead of being square, it's sort of hexagonal. And just like with a millimetric paper that allows you to measure distances, you can use this hexagonal sort of millimetric paper to measure distances of the animal travels to space. So we have in the brain neural maps, place cells, neural compass, neural distance meters, grid cells, and also neural compasses. They're called head direction cells. So these are neurons that don't care so much about where is the animal in space, but they're active strongly whenever the animal is heading or looking in a particular direction. So in this case, this is a neuron that is the head direction cell that's active whenever the animal looks due east in a polar representation. So we have in the neural maps, neural distance meters, and neural compasses, all the elements that you'd like to build a navigation system in the brain. And now what we ask in my lab is, how does real-life navigation differ from navigating in a 1 by 1 meter empty box? Because these the experiments that I just showed you, the examples I showed you, and the typical experiments in the field are done in animals running in these empty small boxes, or maybe on a linear track that's 1 meter in length, and so on. And you can give several answers. So first of all, the world is not flat, it's complex and two-dimensional. How does that affect the system? The world is not empty, it contains goals. How are goals represented in the brain? The world is also not 1 meter in size. Animals navigate large distances, including rats, and certainly other animals. So how are large spaces represented in the brain? And also animals often navigate in groups. So how are the social factors come into play in the system? You can give more answers, but these are things that we are directly studying experimentally in my lab, and this is also the outline of my talk today. So we study this not in rats, but in bats, and I'll actually leave to the end to explain why bats. The idea, of course, is not to understand the bat per se, although they're really cool animals, but to understand the neural base of behavior more generally. But as you'll see, you can use bats to ask questions that are sometimes much more difficult to study using the classical animal models in neuroscience, such as rats or mice. And so these are pretty big bats, Egyptian fruit bats. They weigh 150 to 180 grams, so they're much bigger than the bats that you might see around Konstanz or in North America, which means that you can put a lot of weight on them. You can still need to miniaturize a lot of the electronics, but it helps to have a much larger animal. They can carry a lot of weight. Also, they have, just by looking at their eyes, which are definitely enormous on this screen, you'll believe me that they're not blind. Blind is a misnomer. No bat species is blind. These bats actually see very well and also have a sense of echolocation that I won't have a time to talk about today. And so to put you into the mindset of what does it mean to navigate for a bat, this is from a study we've done some years ago where we put little GPS data loggers on the bats and released them in their cave in Israel, about 40 minutes drive from the Weizmann Institute. And you see an example of the trajectories of one bat that flew from the cave to its favorite fruit tree night after night. So each color here is a different night for the same animal. And you see that they have, this bat definitely knew where it was going. It came night after night to the same individual tree. There is some variability here, so this flyway is not perfect. It has a width of maybe 1 or 2 kilometers, but still pretty good. These animals definitely know how to navigate, and eventually we'd like to understand how they do it at the neural level. So I'll start with the questions of 3D. How is 3D space represented in the brain? Before that, just to assure you that in 2D we found similar things to rodents. So when we have bats, we sort of ratify the bat. We have them crawl in 1 meter square boxes on purpose so we can compare to rats. We found the same kind of neurons, place cells, grid cells, and head direction cells in bats as in rats. And then when we went to 3D, we found Michael Yartsev, when he was a PhD student in the lab, he found 3D place cells. So here bats flew in a 5 by 6 by 3 roughly meter flight room. And he found neurons that look about like this one. So this is a place cell in the hippocampus. And what you see here, that whenever the bat is flying through a particular volume in mid-air, that's about 80 by 80 by 80 centimeters in size or so, kind of a spherical volume, this neuron gets activated. And the fact that it's spherical means that the resolution is the same for the X, the Y, and the Z dimension. So the resolution is the same in all directions, which was under debate whether it should or shouldn't be the case. So at least in bats, flying mammals, this is the case. Then Arseny Finkelstein, a couple of years later, asked, are there neural compasses in the bat's brain? And the answer is yes. So three-dimensional head direction cells that fire whenever the bat's head is pointing in a particular three-dimensional direction, like there, or there, or there. And there was a little twist that I won't have time to go into, that we were able to show in this study that the 3D head direction cells, these 3D compasses, were not operating in a spherical coordinate frame, which is the classical textbook coordinate frame for representing three-dimensional directions, but in what's called a toroidal coordinate frame, which is the coordinate frame for those of you who have an iPhone and you have a 3D compass on your iPhone. That's a coordinate frame used by the iPhone. So it has some interesting mathematical advantages, but I won't go into that. And just to say that following our studies, several other groups found similar representations also in other animals. So Dora Angelaki's group has found 3D head direction cells in monkeys, obviously very much two-dimensional animals, and also in mice, which are excellent climbers. For those of you who have seen mice climbing, they're really great climbers. And also really recently, a few weeks ago, Kate Jeffries' group has posted a preprint on bioRxiv that shows 3D place cells very similar to those. So really spherical or isotropic 3D place cells. So it's not just in bats. Okay, what about 3D grid cells? So this is the work by Gili Ginosar, a PhD student in the lab. So in 2 dimensions, in rats, this is how a grid cell, your kind of paradigmatic best typical grid cell from the Moser lab. This is how it looks. So it's really a nice kind of hexagonal lattice. And it's characterized by two properties, this hexagonality, this famous shape that gives its nice symmetrical properties and the ability to measure distances. And also, it has these fixed distances between nearby fields. And you'd say, wait a minute, it's sort of the same thing. If you have an hexagonal lattice, you also have fixed distances. So why dissociate the two? But in fact, as I'll show you in a second, when you go to 3 dimensions, these two properties are actually not the same. So it's important to look at each one separately. So what might we expect to see in 3D? So here charted some hypothetical possibilities. We might find kind of vertical columns, kind of degenerated z-axis, but a 2-dimensional grid. We might find some lattices, like hexagonal 3-dimensional lattices or cubic lattices. We might find complete disorder, or we might find something which is between order and disorder. So these are the kind of possibilities that we wanted to consider. And so to do this, what Gili did, she had bats flying in the same flight room, 5 by 6 meters, and used a wireless electrophysiology system, so a neural logger, a very small device that weighs just a few grams that allows recording neural activity, the activity of single neurons in flight. I'll show you a picture of that later. And while she was recording in the medial entorhinal cortex, while the bats were flying in the flight room, and you see that they covered the 3-dimensional space quite well and flew quite long distances, 2 kilometers per session, so got a nice coverage of 3-dimensional space. And here are some examples, 8 examples of neurons that Gili found. So by looking at that, you can clearly see that the activity is sort of blobby, right? You have these distinct blobs, but it's hard to see from just looking at it, is there a lattice there or isn't there? So kind of what's going on? It's hard to tell just by looking. What you can say relatively easily, it doesn't seem like you have vertical columns. Okay, you can rule that out. We also quantified it and showed that it's definitely not, there's no vertical columns here. But it's hard to say whether there is a lattice or not. So to address this, what we did is we looked at distances between neighboring fields. So we can look at this field and the distances from all its nearest neighbors, and then the next field, the distances from all its nearest neighbors, and collect all these nearest neighbor distances and plot a histogram of these distances. So here, for that particular neuron, you see the distribution of distances between neighboring fields, and you see that this distribution is narrow, meaning that nearby activity fields have a fixed distance scale, or rather fixed distance scale. There is a fixed scale to the representation locally, and we can quantify this by a coefficient of variation, which is the standard deviation of this distribution divided by the mean. And for that particular neuron, the coefficient of variation is this black line is very low and much lower than all the shuffle data. So this neuron really has a characteristic distance scale, and 20% of the neurons in the medial entorhinal cortex had this characteristic local distance scale. But then we asked, is there a lattice? Does this local distance scale mean that you also have a global lattice, a perfect lattice in 3D like in 2D? So do you have something like that? And this would have taken me too much time to show you all the analysis, but the short answer is no. Okay, we've done a lot of different analysis, and the short answer is that we did not find any neurons that showed FCC or HCP lattices, which are these perfect 3-dimensional lattices that were expected and predicted by several theoretical papers. And also, we looked for other non-hexagonal lattices, like a cubic lattice, so 90-degree angles, or any angles that we can find that would be characteristic. And the short answer is there's nothing, okay? So the short answer is that we have a local distance scale, but no global lattice, okay? So we can rule out this possibility. Showed you we rule out the hexagonal lattices. We can rule out cubic or all other lattices. On the other hand, it's not random either, because there is this local distance scale. So we are left only with this possibility, something that is in between order and disorder. Now at this point, some of you might be confused. How can you get local order but without a global lattice? So just to give you an intuition, here you have a perfect lattice, or almost perfect for you, okay? And how do I know it's a perfect lattice? Because when I look at this structure, it's a perfect copy of this structure. How do I know it's a perfect copy? Because I made this one in PowerPoint and then I copied it over here. So I know it's an exact copy. Okay, so it preserves scale and orientation. So this is sort of the definition of the lattice. On the other hand, this critter, it's clear that there is no, it's not a random structure. You have locally fixed distances, but also when you go from here to there, you see that the pattern rotates. So you have a local structure, but not a global lattice. So this is just to give an intuition how something like this might be possible. So to understand this more quantitatively, we teamed up with a team of theoreticians, Chaim Sopolinsky, Ram Barak, and Yonatan Al-Khadef, whose idea was to look at, to try to model what's going on in 2D versus 3D, and why is this difference between 2D and 3D by looking at pairwise interactions. So the idea comes from models in physics and chemistry, where if you have particles, you can do a lot of, and also Ian done a lot of work along these lines. You can do a lot by modeling pairwise interactions between particles, between animals, between all sorts of things, and get all sorts of interesting collective behaviors. And depending on the regime of the pairwise interaction, you can get things that look very structured, like a hexagonal lattice, or things that are completely random, or things in between. So the idea was to try to use these models from physics to try to understand why is this difference between 2D and 3D. And so this is the particular model that they've used. It's called the Leonard-Jones potential. It has this particular shape, and it's characterized by having, so this is kind of the potential energy versus the distance between the particles, or the fields in our case, and it has repulsion at short distances, attraction at longer distances, and it has kind of a perfect distance, characteristic distance, which is the minimum energy, but there's also noise or temperature, which makes things deviate from perfect structure. And using this model, they were able to show that the data, this model explains the data much better than a random Poisson, a random arrangement of field. This is the distance between the data and the model, and the pairwise interaction model, this particular model, is much, much better than the random Poisson model, and also much better than several other models that we compared to. Okay, so this really seems to capture the data quite well. Now, what happens, why is this difference between 2D and 3D? Can we recapitulate this in the model? So here what they've done is when kind of looked at the regularity index, which is an index of hexagonal structure, and in three-dimensional room, the room which actually has the sizes of our flight room, the regularity index is 0, meaning there's no hexagonal structure. And then as you kind of squish the room from 3D to 2D, so you still keep it at 3D structure, just instead of having a several meter height, you squish it down to 10 centimeter height. So it's a three-dimensional structure, just very flat, like a pancake. And then under these conditions, you get a high regularity index and hexagonal-looking arrangements of fields. This is simulations, yes? And you see here there's a clear phase transition from no global structure in 3D to a nice global structure in 2D. And another intuition of why is this difference happening comes from approaches in physics that looked at jammed packing of spheres. If you think about, if you throw a lot of spheres here on the floor and kind of pack them together, you'll invariably get a hexagonal structure. But if you throw a lot of balls into a three-dimensional pool, like a jamboree pool in a ball pool, you'll never get a perfect lattice. You will, on the other hand, get local distances, because all these spheres have a distance between each other that is the diameter of the sphere. So they're all sort of touching each other. Okay, so you have a local structure, but not a global lattice. So this is another way to get an intuition of what's going on here. Okay, so to summarize the story on 3D grid cells, we found cells with multiple 3D fields. 20% of them are grid cells, which have, or even 30%, depending on the measures and how we do the shuffling, which means they have a local distance scale, but none of them, not a single one, had a global lattice. So that means we have a local but not global metric of space. And we suggest, therefore, that the fundamental property of grid cells is having multiple fields with a characteristic pairwise distance. And in 2D, this model yields a global hexagonal lattice that emerges from pairwise interactions confined to 2D. Okay, you confine pairwise interactions to 2D, and you'll get hexagons no matter what. It's just an emergent property. On the other hand, in 3D, the exact same model, the exact same equation gives you a local structure, a local metric of space, but no global lattice, exactly as in our data. So the same model explains both 2D grid cells and 3D grid cells. All right, let me switch gear and tell you a little bit very briefly about representation of goals. So far I told you a little bit about representation of position, representation of distances, but in fact to navigate means to get from point A to point B. And everything I told you so far has to do with point A, right? Place cell tell you are here in point A. Grid cell tell you something about local distances around point A, but where is the goal? I can imagine, some poster boards that are out there. I don't see them, but I know there are poster boards out there. So how is this represented in my brain? Where is point B in the brain? This was unclear. So what Ayelet Sarel, a PhD student in the lab, did is exactly to pursue this question. And just to show you that bats can know very well the positions of goals, this is from recordings that we've done with Yossi Ovel. Some years ago, we've GPS tracked bats, and this is one animal that is sort of the champion, was tracked for 75 nights in a row. I'm just showing you part of the nights, not all of them. It becomes pretty dense. So this particular bat flew from the cave to this tree for many nights. Then it switched to this other tree. And then on night number 33 in Cyan, it started flying in the direction of the first tree and then changed its mind and went to this other tree. And there are also other nights when the bat did shortcuts from this tree to the other or to another tree that's located there. So really very interesting behavior that it's interesting to look at. But the point of this is just to say that they know and they represent goals. So how can that work in the brain? So Ayelet hypothesized that one simple way to represent goals would be just to represent the direction to the goal. The nice thing about direction, azimuth, is that 360 degrees are the same 360 degrees on all scales, whether you work on this scale or in a small room. So this would be a nice compact representation of goals. So she went to study this by having bats fly in the same flight room, 5 by 6 meters. This is a top view. And all she did differently from the typical experiments that have been done in the last 40 years in the field is that she introduced a goal in the middle of the room. That's it, the only change. So it's really very trivial. Put a landing platform in the middle of the room. And so the bats were flying around and doing these various trajectories. As you can see here, these individual trajectories flying out and then after a complex trajectory landing back in the goal and again and again and again. And this allowed us then to look at this angle, which we call the goal direction angle. It's the angle between the direction to the goal and where I'm heading. So if this podium is the goal, is my goal, so when I'm heading in this direction, it's minus 45 degrees to my left, and if I'm heading towards it, then the goal direction angle is 0. Okay, so you can really look at the angle relative to the goal. And recorded in the hippocampus of the bats. And what you found that there were indeed neurons that represented this variable, the goal direction angle. So what you see here is the activity, the firing rate, the rate of these spikes, of this action potential of one neuron versus this angle, the goal direction angle. And you see that this neuron has a nice tuning with a peak at direction 0. This means whenever the bat is flying towards the goal, direction 0 is towards the goal, then this neuron is active maximally. So by looking at the activity of the neuron, you can tell the direction to the goal. Likewise for this neuron. And there are also neurons that were tuned, but to a non-zero direction, like this neuron that's actually active maximally when at 180 degrees, meaning when the goal is behind me, okay? And different neurons have different preferred direction like this, but a very large fraction of neurons in the hippocampus represented the direction to the goal in this way. And then we asked also, are there neurons that are not only representing the direction to the goal, but also the distance to the goal? So here what you see is the firing of one neuron as function of the goal direction at distance 0. So it's nicely tuned for goal direction 0. But then when you go sort of away from the goal, minus distances here away from the goal, you see that this neuron is still tuned to goal direction, just the firing rate goes down. So essentially you have here a sort of two-dimensional tuning curve. This neuron represents both the direction and the distance to the goal, okay? And the direction and the distance has a name, it's called a vector. So essentially these neurons represent the vector to the goal. So to summarize this story, we found in the hippocampus neurons that represent goal direction. We did experiments that I didn't have time to show you where we put curtains and the bat had to navigate to a goal that it knew mentally where it was but couldn't see it, and the neurons still represented the direction of the unseen goal. So this means that this representation is memory-based or mental, just like me knowing where the posters are, even though I don't see them. And together, these representation of direction and distance forms a vectorial representation which could directly support goal-directed navigation. By directly, I mean this gives you the information that you need to exert action, right? This tells you where your goal is. So all you need to do is to go to the direction of the goal, right? So it's as close to the action side of things as you can get. And also, this might be the neural basis of the home vector. So the home vector is a concept that goes back to the 1950s or maybe even earlier. And people have shown in many studies, from insects all the way to humans, in many animal species behaviorally, that animals can do vectorial computations when navigating. But this was unknown how in the brain this is done. And these neurons that represent vectors might be the neural basis for that. And just to say that, again, several groups, after we found these kind of vectorial representations in the hippocampus, looked at, did experiments to test this in rodents and found similar cells in both rats and mice. So it's, again, not just in bats that these kinds of neurons are found. Let me now switch to talking a little bit about social representations. So this is a work by David Omer, who just last month started his own lab as an assistant professor at the Hebrew University. So what David asked is seemingly a very simple question. In the hippocampus, this brain area that represents where I am in space, are there also neurons that represent where you are in space, so the positions of other individuals? So bats are highly social mammals, so it would make sense that they would have a representation of the position of other individuals. We know behaviorally it's very important for them to know where are the other animals. So we decided to look for such representation in the hippocampus. And to do that, what we came up with is a task that we call a delayed match to place task. A very simple task, but that's very useful for asking this kind of questions. So it involved two bats in this case, the blue bat, which was the demonstrator, and the red bat, which was the observer bat and was also implanted with tetrads in the hippocampus. And so the blue bat, the task of the blue bat was to fly randomly for the start ball to ball A or B, and then back. So in this case, it flew to ball A and then back. And then the task of the red bat is to mimic it. So the blue bat flew to A, so the red bat also flies to A. If the blue bat flies to B, the red bat also has to fly to B. So here you see that this task has several interesting features. First, attention. The implanted bat, the red bat, has to attend to the location of the other bat in order to do the task. So we know it has to pay attention to the location of the other animal. Then we have a hope, at least, to find a representation of the location of the other animal in the brain. And the second is this what we call a behavioral space clamp. You noticed in this cartoon that one bat was flying and then back, and then the other bat was flying and then back, and they were not flying together in the air. The reason we did it this way is because this is the hippocampus, and if both bats are flying together, this becomes more tricky because there are place cells, to represent where I am in space. So it's more tricky to disentangle self from other. So that's why we did this task where when other bat is flying, I'm not moving, so we can disentangle it. And in addition, we had an accelerometer on the bat's head to make sure to rule out the possibility that any miniature head movements could explain the response to the other bat. So I won't show you this control data, but we've done all of that. And so this is an example of what we call a social place cell in bat hippocampus, bat CA1. So what you see here, this is all from one neuron. On the left is the classic representation for cell flight. So here the gray trajectories are the flight of the bat itself, the implanted bat. And you see that it's flying to A and to B, and there's not a single red dot, meaning that it fired absolutely zero spikes when it was flying itself, meaning this is not a place cell, okay? It is not firing when I'm flying. On the other hand, when you were flying, when the other bat was flying, this neuron fired clearly on the right-hand side, encoding the position of the other bat. Okay, so we call these social place cells representing the position of the other bat. There are, of course, also classical place cells like this one that were active very nicely when I was flying, but did not fire at all when the other bat was flying. And there were also neurons that did both. In this case, this neuron fired on the left when I was flying and on the right when the other bat was flying. And in some cases, the activity was in the same location, so the same position for firing for self and other. In other cases, they were different, like in this neuron. Couple of controls that we had to do. One thing you could say, well, maybe it's not really representing the position of the other bat, but planning of my own movement, right? I'm looking at the other bat while I'm thinking or planning what I'm going to do. So it's really neurons that represent planning. So we did 3 different controls to rule this out. I'm just going to show you one. So the idea was to look at correct versus incorrect trials. You'd expect that in an incorrect trial, the other bat flying right and I am flying left. So that means there's a difference between what the other bat is doing and my planning. And then you expect a difference between correct and incorrect trials. But in fact, we found not much of a difference. These are examples of 2 neurons in correct top and incorrect trials at the bottom. And you see that there is a similarity in the firing fields for correct and incorrect trials for these 2 neurons. And when you look at the correlation between the maps for correct and incorrect trials, the correlations are high. So this rules out this idea of trajectory planning. And lastly, we asked, well, maybe it's not really social. Maybe you don't have to have another bat to evoke these responses. Maybe anything that moves out through such an activity field will evoke a response. So to do this, we had, in addition to this task to this session of following the other bat, we had 2 additional sessions. One is follow the object. So we had a football, literally an American football, like on a stick, move to either ball A or ball B, and the task of the bat was to mimic it. So it's the same task, just you're mimicking a plastic football rather than a live animal. And then we had a 3rd session where the bat was not supposed to do anything. There was an object moving out there, a cube, but the bat was not supposed to respond to it. And I'm just showing you one neuron. This is all one neuron. And you see that it in fact did respond to these 2 objects, what we call the informative object, the football, and a non-informative object, the cube, in a similar way to the 2 objects, and in a differently than to the other bat. And invariably, this is what we found across the population of neurons, that the representation of the live animal, the other bat, was always different than the representation of the objects. So we conclude that our results demonstrate that the position of other individuals is explicitly represented in the mammalian brain. And in parallel to us, similar results were also found in rats by Fujisawa's group in Japan. And the representation of a conspecific is different from the representation of moving objects. And we think that social place cells may be important for social spatial cognition in mammals, representing conspecifics, which is what we've done here, but maybe also other important things like predators and prey, etc. And we are now doing experiments. Psychiatry, a postdoc in the lab, has set up this room where we have 10 bats living together. We develop methods to track the positions and identities and head directions of 10 individuals flying and moving in a 3-dimensional, cluttered, complex environment. And now with this wireless neural loggers that we have, we can record potentially from all these 10, the brains of all these 10 animals simultaneously. Okay, we're not starting from all 10 bats, we're starting 1 or 2 at a time. But because there are no wires dangling out there, we can, in principle, record from all of them simultaneously. What we want to look at are things like, is there a representation of identity? Okay, are these social places responding differently to bat A versus bat B? Is there a representation of males, a different representation for males versus females? For dominance hierarchy, for friends, because these bats are really interesting. I don't have time to go into this. They really have very interesting social interactions that are individual specific. They would interact in one way with one individual in completely different ways with another individual. So are there different representations for the friends, the bats that I like to hang out with? So these are all questions that have not been asked in general in neuroscience, but we are asking them in the bats, and experiments are conducted. Neural recording took us a year to set up the behavioral tracking in 3D for the bats, but now it's all working. So Psychiat is now starting implanting bats, and we hope to have some answer to these questions very soon. And lastly, in the last part of last few minutes of my talk, I'll tell you very briefly about representation of large-scale spaces in the brain. This is work by Tamir Eliav, PhD student in the lab. So the basic question or problem, if you will, is illustrating if we go back to this slide. So if you want to take, you have here the flyway I showed you before. So it has a volume of about, the distance here is about 15 kilometers in this particular case. And it has a width of 2 kilometers and a height of about a half a kilometer. So the bats fly from an altitude of about 100 meters to 600 meters or so. This is what we measured. So we have here a kind of a corridor that's 15 kilometers by 2 kilometers by half a kilometer. If you want to tile it using laboratory scale place fields that you measure in a 1 meter box that are about 10 centimeter in size, then you can make a little calculation in the back of the envelope. And what you'll find is that you need about 10 to the 15 neurons in order to tile this kind of volume. Okay, the entire dorsal hippocampus, this area where place cells are located, has 10 to the 5 neurons, 100,000 neurons. Okay, so we are 10 orders of magnitude off. This means there's no way this is going to work. There's no way that what we find in small boxes in the laboratory will translate one-to-one to what's going outdoors. It's just impossible. It's 10 orders of magnitude off. It's not going to work under any circumstances. So there must be something different. And this is what Tamir is after. And they also fly these long distances in underground structures like this one. Is this cave system. It's a maze-like cave system where they fly hundreds of meters before flying out. There's a little colony of bats on the right side of this cave system. And so to answer this kind of question about representation of large-scale space, we had to solve 3 problems. One is, how do we record neurons over large spatial scales? The early recordings that we used for the 3D place cells were wireless, but they were telemetry recordings, meaning we transmitted the data. That works for a distance of a few tens of meters in the lab. That will never work over long distances. So for that, we developed this neural logger that stores the data on board the animal. So now we are unlimited in distance. And this is the kind of the first generation of these 16-channel neural logging system that allows really to record very nice spikes. This is the first of the 4 channels of 1 tetrode. And these are the spikes, these electrical pulses of 1 neuron in the hippocampus. You see these nice spikes. For anybody who's done electrophysiology, this is supposed to impress you. It's a really nice signal in flight. And we have just transitioned to a 64-channel neural logger, meaning 16 tetrodes, which allow us to record now neural ensembles, large groups of 50 to 100 neurons simultaneously in a freely flying animal unconstrained. So this is one thing that we had developed. The other problem that we had to solve is the positioning. If we want now to differentiate between a 10 centimeter precision, your 10 centimeter place field in the lab, and something else, we need to be able to measure the position of the animal with 10 centimeter precision over long distances. Now, GPS is not good enough. GPS is about 10 meter precision or so. And there are now kind of RTK solutions that we are moving to that allow to have much better precision, but let's say classical GPS is definitely not good enough. And the reason GPS is not good enough is that it measures distances to, it works by measuring time differences of arrival to satellites in outer space. And on the way, there's the atmosphere, which is great for us, but it also problem for localization because there are atmospheric aberrations that distorts the signal. So we found this solution from a French company that has something similar to GPS, but instead of satellites in outer space, we deployed an array of ground-based antennas locally, and this then allows us to measure these distances, these circle, and intersect them, and this gives the position of the animal to about 10 centimeter precision, or even a bit better, 8 or 9 centimeter precision over long distances. Okay, and the third thing that we need to develop, we want a controlled setup, but a very large one. So we've built this tunnel in the Weizmann Institute campus on the east end. It's 200 meter long, so you see the very end of it, and we put landmarks in there, and then we have the bats flying in there, and they fly like hell in there. So this is 200 meters, this is time, and the bat is basically flying. We put food at the 2 ends of the tunnel, and the bat is flying back and forth and back and forth and back and forth and back and forth and back and forth. And initially when we were building this thing, I was worried that, how do we convince the bats to fly there? If we release a bat and it doesn't fly, what are we going to do? I was telling you, run after it, we're going to buy segways to drive after the bats, and what are we going to do exactly? But the first time we released bats there, all the worries were evaporated because these are marathon flyers, and when we give them these enormous distances, they do what they like, which is fly a lot. And they fly 100 or even 120 laps per session. This means 20 to 25 kilometers in one recording session, which is an enormous distance. Also at very high speeds. In this case, the speed you see is very fixed, and it's very high, 7 to 8 meters per second. And some days the speed is 10 or 11 meters per second. That's 40 kilometers per hour. That's very close to what they do outdoors. And so here we're utilizing the bat's flight speed to measure the representation of large space. Yeah, if you release a mouse there, it'll do a couple of laps and say, okay, I had enough for today, but this is the flying animal, so you can fly 100 times this distance. And here is an example of a neuron that Tamir recorded in this environment. So all of this is one neuron. What you see here is a raster. So each line is one flight, and dots are spikes, and they're separated into the two flight directions, the red direction to the west and the blue direction to the east. And what you see here are several things. First of all, you see that the representation of the flight to the west and to the east, the blue and the red, are different. Okay, so that's known already in the hippocampus, that on the linear track, the representation of two directions is different. Think of yourself going from home to work or from work to home. This sort of feels different. So that has been known for many years. What's new here are several things. First of all, that this one neuron is active not in one location. I showed you before place cells that are active in one location. It's kind of a paradigmatic Gaussian firing field. But here it looks completely different. This one neuron is active in many locations, here and here and here and here and there. So multiple locations. Also, the sides of the place field are huge. Here you see this place field is about 20 or 30 meters in size, and that's in dorsal CR1, where you're supposed to have these 10 centimeter place fields in small environments. So there's definitely scaling up when you go to a large environment. And lastly, and most surprisingly for us, this neuron has multiple spatial scales in different locations. So it has a large field here and a small field there. And here is another neuron that demonstrates the same phenomena. You have multiple fields, and the same neuron has a large field, meaning a low resolution in this location, but a small field, meaning a high resolution in a different location. Okay, so these neurons have large fields, multiple fields, and this multi-scale property, which is extremely surprising. There's not a single model of place cells that can explain how the same neuron can have different resolutions in different locations in the environment. And it definitely looks very different than what you find in small laboratory environments. And we are now working on understanding the mechanism, is that possible, and also the function. What's the computational advantage of having such a multi-field, multi-scale code? We have some ideas. And we're also building on the Weizmann campus almost 1 kilometer long flight tunnel and a very large maze, about a football field in size. In the tunnel, we can look at the representation. This is also under construction, much like Ian's buildings. So a lot of excavations right now on the Weizmann campus. So we have, in the tunnel, we can look at the representation of multi-scale environments in the brain. So compare a large environment to a much smaller environment. In the maze, we want to look at questions like neural codes for complex environments. There's very little work has been done on how just a complex environment rather than an open box. How is the complex environment represented in the brain? Also, what happens when you have multiple goals? We can put multiple goals here, 4 or 6 goals in this environment. How are multiple goals represented in the brain? Each one of us can point to multiple locations in Konstanz, at least when you know the area. And lastly, what happens in the brain when the way is blocked? When a mischievous PG student blocks the way and the bat has to reorient, recompute its directions. What happens? Is this vector literally rotating in the brain? So questions of that sort we can directly address in this maze. Okay, so the chief take-home message that I want to leave you with is the following, is the importance of looking at the neural base of natural behavior. So I'm sure here it's preaching to the choir, but the idea is that, Western science in the last 400 years, we've been all taught about the importance of reductionism. Okay, I'm a physicist by training, and when you're studying, the hydrogen atom, you'd like to study it in a vacuum, in a cold environment, not in some bush somewhere. But when it comes to behavior, what always worries me if we oversimplify behavior and look at the behaviors that are too controlled in the lab, we might be missing the very thing that we are trying to understand, which is behavior, because behavior is complex, it's context dependent, it's rich. Of course, control has its advantages, but I think on the other hand, we also need more diversity. We need, in addition to the controlled experiments, also have the more natural or naturalistic types of experiment. And I showed you from our own work that every time we'll do a little baby step in a more natural direction, we find surprising things. We go to 3D instead of 2D space, we find the grid cells look completely different, toroidal, head direction cells, etc. We introduce a goal in the middle of the room, just a landing platform, and suddenly we found vectors in the brain and anticipated in the hippocampus. We introduce another animal, and suddenly we found a representation of the other animal. We enhance the size of space, and all hell breaks loose. It looks completely different than found in small environments. So each time we do, none of this is really natural environment fully, but still it's little steps in more naturalistic direction, and things look completely different. So I think we need to have more natural behaviors in behavioral neuroscience. And before closing, I'd like to plug in a conference that we are having, organizing together with Yossi Oved and Cindy Moss on active sensing, the Weizmann Institute in March 2020. Here's the link, and for those who are interested, it will have a large variety of species and humans and robots, etc. So for those who are interested, you can register here or come talk to me. And so with this, I'd like to finish and thank the lab members, and especially Lior Alas, staff scientist, who is fantastic, really great, and has been working with me for a number of years, and she's been involved in most of what I've shown you today. And these people in the lab, I think I mentioned most of their names as we went along, and alumni and collaborators and funding sources, and thank you for listening. Yeah. Mm-hmm.
I have a question. So in the beginning, you kind of motivated your entorhinal grid cells by saying, oh, it's kind of a metric for odometry or whatever. And now in the bats, it's kind of a local metric maybe, or is that still how you think about it? Or do you think it's doing something else?
So it's a local metric, yes. The idea, it's not a global metric of space, but it's a local metric. So it's still, it's not random. There's still, it means that there's a local distance there, but just, if you think of it, it keeps getting distorted over space. So some of the things that, some of the uses that people have in mind for grid cells can still work with this thing, but some models would fail. For example, models that require precise periodicity globally, like the modular code types of models, all sorts of models that I won't go into, they're not going to work. Okay? So basically, the division line is if a model requires a precise global lattice, then that means it fails.
Thanks. At the landscape scale, how are you ensuring that the firing that you're detecting is purely spatial and it's not in response to some sort of cue, perhaps even...
In which experiment?
At the landscape scale there, where you've got the single neuron firing at a number of different locations.
Oh, you mean the last part,
yes,
the multiple fields. Okay.
How are you sure that that's spatially related rather than cue related, so
right.
there's not some underlying electromagnetic field or sound or visual cue?
Well, electromagnetic field, that's a magnetic field of the Earth doesn't change on scales of a few meters. Yeah. We did, at least for the landmarks that we introduced, we looked at that. I didn't have time to show you, but we looked whether these fields tend to be located near landmarks, and the answer is no. Mm-hmm. They really seem to be almost uniformly spread around space and not cluster around landmarks, or not even that the small fields are around landmarks and the large field are in between landmarks. It doesn't seem to be the case. We looked at that. Okay. So they definitely seem to look way beyond a landmark and to really know their position. But it does have a very interesting implication about the code, because that already means automatically that the decoding, it has to be a population code, and the decoding is perhaps not what we thought. But it doesn't seem to be responding to specific cues.
Thanks.
You're sort of describing the evolution of bats, or the representative of space. So bats started in the oceans, like we all did. Then they moved from a three-dimensional space to a two-dimensional space when they moved to land, and then they returned to a three-dimensional space. So the idea that the original representation was a three-dimensional representation, and then it was squished when we moved to land, and it was re-released when the bat took the air, evolutionarily speaking?
So, well, it's a good question. We know very little. So to really answer this, we need to look empirically, record neurons outside mammals, which people are now starting to do. So actually,
there is some fish work now starting. There is some fish work
in the fish,
now.
exactly. So Ronen Segev at Ben Gurion University is actually using our neurologgers. He found a way to seal them against water. Of course, water and electronics don't work very well, but if you seal them, it does work, and he's able to record from fish. And he is finding the strongest representation that he's finding are what's called border cells. So neurons that are firing along the tanks, the borders of the water tank, which I find great because it's literally, for the fish, it's literally the end of the world. It's as strong as the border as it can get, and neurons really seem to care about that. I'm also collaborating with Joram Gutfreund and the Technion to work on
Barn owls.
in barn owls and also quails, so to look at in birds. And he's seeing all sorts of interesting things. So I'm certainly very much for looking outside mammals as well. And so it's hard to for me to answer your question. What I can say is that so far, a lot of what we found in the bats initially was also later found in rodents, which you'd think are much more two-dimensional, but in fact are great climbers. So both the 3D head erection cells and the 3D place cells. So 3D head erection cells was published already. The 3D place cells, as I said, was posted on bioRxiv a few weeks ago. And Kate Jeffrey has also results which she told me about. She showed me, it's not posted on the bioRxiv yet, but where she found grid cells that are very much like what we are seeing. The rats are climbing in this three-dimensional kind of mesh, essentially, also with a wireless neural cortex device. And she's seeing a local but not global structure. So it seems to be that it's, at least some of these things are general across mammals, but outside mammals it's really hard to say.
Can I? Hey, so you mentioned that social cells may be able to encode identity or caste or relatedness of the other bats. Do you also observe any difference in the learning performances depending on, the bat, the demonstrator, so whether it's depending on its size or relatedness? And if so, could these cells actually encode something to do with the propensity to perform the task rather than the identity?
Well, we haven't tried that comparison. Yeah, it's a good question. So first of all, all our experiments are done when the animal is well trained. So we haven't really looked at as the learning evolves. We are starting to look at this now. So now that we have the 64-channel neural logger, we can record many neurons simultaneously, so we can record every day a lot of neurons. We're starting, in the tunnel, for example, we have a student in the lab, Shir Maimon, she's now recording in the tunnel from day one, from the very first day the bats are exposed to see how the map develops, things of that sort. And then we can start asking questions like you're referring to, so the learning, let's say, in a social task. Well, we haven't done any of that. All the recordings that we've done, after the animals are all very well performing.
Hey Nachum, thank you for your great talk, as always. I was wondering, most of those tasks that the bats have to perform are pretty straightforward and actually cue-oriented, whether it's not actually a hippocampal activity that drives them, but striatal. Have you considered that?
No, I totally agree with you. This is part of the, I would say, the mystery, because place cells, also in rodents, you find them always, no matter what, also when the animal doesn't have to pay attention to it. Like when the rat is randomly foraging in a box for thrown food pellets, what it cares about is the food pellets, not its location and environment. And still, you always find place cells, right? So it seems to be a very strong representation that's always there. Another thing that I didn't have time to go into, we have in the social task, we have now new results on what we call episodic cells. So these are neurons when the bat is sort of doing nothing, we find time cells, so neurons that are firing in a particular moment in time when the bat is sort of hanging there and doing nothing. And these time cells are activated differently in the different locations in the room. And so they're encoding simultaneously time and space-time, if you will. And not only that, we find that these neurons are presenting not just time and space for me, but also time and space for you, for the other bat. Okay? So great for anybody who likes episodic memory and the like. But you could ask the same question about that, right? There's no time component in the task. The bats don't have to do anything, lick or respond at a particular time. They don't really care about time. So why should there be a very robust time representation? Almost 50% of the neurons have these time responses, very dramatic. Still, it's always there. So it seems that at least time and space, and so in this sense at least O'Keefe was right, I think, in the sense that time and space, these kind of Kantian variables, are very strongly represented in the hippocampus no matter what. They're probably just very important to be represented always.
Okay, cool. So we're gonna cut questions off here, and I'm gonna give you a brief update, and then we can enjoy some drinks down amongst the posters. So let's give them another round of applause.
Thank you.